So I mentioned bleaching, I mentioned boiling, I mentioned UV damage. You have here a hypothetical virus, no particular pedigree, with a different style of virus. This is a retrovirus, which works a little bit differently. Instead of injecting DNA that's just flagged to be used, it injects RNA and retrotranscriptase enzymes, which once they get into the host cell, copy the RNA to DNA. In a healthy living animal cell, plant cell, whatever, even bacteria, or amoebae or fungi, let's not leave them out. We're very inclusive here. You have DNA with genes in it, and transcriptase copying the DNA to an RNA sequence, messenger RNA sequence, which is then used by the ribosomes as the code from which to, the code that encodes, God that's scruffy, a protein, for example. Here you have retrotranscriptase, which copies RNA to DNA, which then hijacks the cell. We've got your protein capsid, just the shell, and we've got a bit of stolen cell membrane from the previous host cell, with proteins sticking off the outside. These are the receptor proteins that will recognise, best word for it, surface proteins of the next host cell and allow this to infect them. So what can we do to this to get rid of it? Well, one thing we could do, we could leave it alone, because eventually it's going to fall apart. How long does this take? Well, for one currently relevant virus, a few hours in contact with copper. Apparently copper's pretty reactive stuff. Copper must in some way dissolve into this and mangle these proteins or mess up the membrane or something. On steel or plastic, days. On cardboard, I think it's 24 hours. Probably in the soil, not very long at all, because this is protein. If a bacterium comes along that this can't infect, because bacterium, not an animal cell, doesn't have the right surface proteins, it bumps into this. Hey, protein! Om nom nom. Just digest the whole thing. Great. Hey, look. Ribonucleotides, I'll have them. Ooh, protein. Amino acids, I'll have them. Hey, all these proteins, more amino acids, I'll have them. Om nom nom. Virus gets digested, problem solved. I wouldn't want to count on it, but I suspect that contact with a healthy compost heap is less able to transmit a virus, or able to transmit a virus but for a lot less time than contact with, say, a plastic surface. Let's talk of bleach. Without going into the actual molecular reactions, bleach is nasty stuff, it'll mess stuff up. If you bleach this and it messes up all of these proteins, well, that can no longer infect anything. Or if bleach is strong, good enough to get through the membrane and mess up all of these proteins and get in here and mess up the RNA, or it's a small enough molecule with the right solubilities to just go right past the membrane and the capsid and mess up the RNA, pow. Doesn't matter whether these are still active. It doesn't matter whether these are still active. If this thing gets to a potential host cell and shoves in a bunch of retrotranscriptase and a few scruffy bits of ribonucleotide, like 8 bases long, with no genes left intact in it, it's not going to cause an infection. There's always fire, just incinerate the thing. Whoosh. You can wreck pretty much anything with fire. You know, burn it to ash. That'll disable it. Ultraviolet light. This is a great way to sterilise drinking water. Sterilise, not filter. This is an important clarification. If there is arsenic or cadmium or hexavalent chromium or strontium-90 or polonium-210, uranium-235, hydrogen cyanide, ricin, botulinum toxin, lead, mercury, ethanol, methanol, anything like that, sulfuric acid in the water, this won't do anything about it. If there are tiny bits of razor sharp, broken glass in the water, this won't do anything about it. But if you have something very much like a child's toy marble race, crude illustration, not meant to be indicative of actual scale or anything, in which water flows in there and has to flow back and forth along these and eventually flows out down there. And you have this thing on a very shallow slope so that the water is going to flow quite slowly down it, and it's reflective, it's a mirror base, and these are clear or mirror shelves, and it has two mirrors over it as a roof, and in here you have a really bright UV light of a wavelength that's strongly absorbed by DNA and RNA. Just give it multiple wavelengths. A lot of lights go off multiple wavelengths. As long as that light is lit and the water, the whole time water is going through here, so you have a timer so it stays lit for 30 seconds or however long it takes after you turn the tap off. Going back to that flask of bacteria and that graph of bacterial population over time, if you take one of those samples where it has 100 million colony-forming units per cubic centimetre, and you put 1 cc in each spread out in each of several empty petri dishes, and put them under a UV light for 5 seconds, 10 seconds, 15 seconds, 20 seconds, and so on, and then actually just spread it straight onto the Newton agar gel and put it straight under the UV light. When you get obviously you have to have the correct dilution as well, or correct dilutions. When you work out do it with the empty petri dish. So I'm working it out as I go along. Do it with the empty petri dish and take samples out of that and dilute them. When you work out how many colony-forming units you have left, you may find that after 5 seconds, instead of 100 million, it's only 10 million. After 10 seconds, instead of 100 million, it's only 1 million. After 15 seconds, instead of 100 million, it's only 100,000. After 20 seconds, 10,000. After 25 seconds, 1,000. However many seconds it takes to wipe out like 99% of them, the ones that are left are probably mutants. This is used in labs to deliberately generate mutants, so you can study mutations. Well, if you do that here, if you know that from here to here takes long enough to wipe out 90% of your colony-forming units, 90%, 99%, 99.9%, 99.99%, 99.999%, you give this thing enough length and enough sloping shells in it to wipe out 99.9999999% of the colony-forming units, the water that comes out of the bottom is very unlikely to be infectious, no matter how many live cholera bacteria went in at the top. What came out of the bottom can contain cholera toxin, it can contain lead chloride, it can contain all kinds of nasty stuff, but it won't contain live cholera bacteria. Not in large enough quantities to set up an infection. Same could apply to viruses. If you have a waterborne virus like poliovirus, which causes poliomyelitis, used to be spread by swimming pools. Oh yeah, they closed the pools in a big hurry when there was an outbreak. The same thing applies. Do enough damage to the RNA or DNA within the virus on its way through, by the time it comes out the bottom, the odds of there being a viable infectious particle left in even 10 litres are tiny. So that works. That's great. What else could you do? You could boil it. Anyone's ever boiled an egg, or had an egg boiled for them, made an omelette, whatever, is probably familiar with the fact that egg white goes from clear and runny to opaque and solid when you cook it. Why? Because proteins, as in these things and these things, get denatured by the heat. Biological washing powder. You use it up to 40 degrees centigrade, you don't use it for 80 and 90. Why not? Because it's enzymes, they are proteins, they get denatured by heat. So boil the hell out of your water and you denature enough proteins, you kill the bacteria. Boiled water becomes safe to drink. Mostly. I mean, again, boiling water won't remove lead, it won't remove potassium cyanide, it won't remove dimethyl mercury, it won't remove arsenic, but you will boil off any methanol that's in it. That's worth doing. So you can burn it, you can bleach it, you can boil it, you can bake it with ultraviolet light. What else could you do? Glacial acetic acid dissolves membranes, wonderful stuff. Great way of getting at the DNA inside a cell. Glacial acetic acid, all the membranes dissolve. That membrane, and these membranes, and these membranes, and the nuclear membrane, and you get the DNA out. So that works. I mean, just take the thing apart completely. These are no longer in a membrane because there is no membrane. They can bind onto animal cells all they like. They're not in any way connected to this. They can't trigger infection. That's good. What else could you do to it? I mean, sodium hydroxide. It's like using acid, but using an alkali instead. It's still corrosive. That'd probably work. Yeah. XKCD has a comment on this, which I should have had on screen ready to read out to you. XKCD cells. Let me just get the wording right. Yes. When you see a claim that a common drug or vitamin kills cancer cells in a Petri dish, keep in mind, so does a handgun. Think about it. You can set fire to the virus or cholera bacteria. You can boil them. You can bleach them. You can soak them in glacial acetic acid. You can soak them in sodium hydroxide, drain cleaner. You can blast them with UV light. How many of these sound like things you want to do to the inside of your lungs? None? I would hope. What happens if you expose yourself to fire, you know, shove your hand into a roaring blue Bunsen flame or a bonfire or something? You're likely to damage the skin. Fortunately, your hand has dead skin on the outside and live skin underneath, and the really important stuff is under that. So you can get away with it very briefly. You can put your hand through a candle flame, which is relatively cool, you'll just get your skin covered in soot. But then you may not have heated your skin, the outside of your skin, enough to kill any bacteria on it in the process. So, dubious whether that would work. Your lungs don't have protective skin over them. So, yeah, setting fire to the inside of your lungs, probably not a good idea. Boiling. Anybody here ever survived being boiled? No? Funny that. Yeah. What happens to people who fall into geysers and hot springs that aren't quite boiling? Generally speaking, they die.